All right, guys, so let's go ahead and start taking a little bit of a look at the nervous system here. We'll just kind of jump in and talk a little bit about some introductory pieces first. So the system can be broken out into two parts. There's the central nervous system, known as the CNS, and the peripheral nervous system, known as the PNS. Central nervous system can be broken down essentially into those two parts, the brain and spinal cord, and then obviously the peripheral nervous system is then anything outside of the central nervous system. Um, Peripheral nervous system, when we talk about that, we're really talking about the nerves and then what are called ganglia that are outside the central nervous system. Each nerve essentially is a bundle of nerve fibers or neuron fibers, and those neuron fibers are bundled essentially by connective tissue. They're much denser than a lot of the other tissue that we see in the body, so they're real easy to kind of tell apart from some other things. So it's pretty easy to see a nerve versus like a blood vessel or a tendon because they're a different substance. Okay, this just shows you real quickly the structure of a nerve. I usually kind of liken them to like electrical wires. So they've also inside of electrical or even speaker wires, they have this kind of bundle of fibers inside of it. So each one of these little pieces is a fiber, and each fiber then would be one cell. The fibers are grouped into a piece that's called a fascicle, very similar to the muscular system that we'll talk about later on this unit, or not this unit, but in another unit. And then those fascicles are bound together by their own little piece called an epineurium. All right. Nerve cells, neurons essentially are equal to the nerve cells. This is usually a question that kind of comes up on a test. What's referred to as the cells inside of the nervous system? They are neurons, and they're specialized in a way so that they can transmit messages as well as receive them. And there are two major regions to them. There's the cell body, and then the processes that extend away from those. So the cell body is usually the nucleus as well as the metabolic center. And then the processes that extend away from that would be the axons and dendrites that we will see here in just a second. So here's your basic nerve. Here's the cell body here in the center. This is the nucleus. All right. These little pieces that extend away from the cell body are the dendrites. These are the ones that are receiving information coming in from maybe another nerve cell or other receptors. All right. This is essentially the nerve fiber itself extending away from that, and then this is the axon out here at the end. So this is what's sending that information either onto another one of these nerve cells or sending it onto central portions of the spinal cord or brain stem. These little pieces that are on here, that's myelin, all right? And that's what kind of helps speed up or kind of make that process take place a little bit faster. So axons end in what's called an axon terminal. And the axon terminal is what actually contains the neurotransmitters that will cross the space that's in between each one of the nerve cells. So nerve cells don't actually touch each other. There's a space in between them called the synaptic cleft. And it's also sometimes referred to as the synapse. So the neurotransmitters are the ones that actually pass across that synapse to go from one nerve cell to the next. All right, here's a little bit more of anatomy. So these are the extensions or the processes. So the dendrites, these are the little extensions that move away, that are out away from the cell body. They are transmitting impulses towards the cell body. And then the axons are transmitting that impulse away from the cell body. So the only way that I kind of keep these straight in my own head is that axons begin with an A, so I think away, and then dendrites don't begin with an A, so they're transmitting that towards. You see some of these other pieces I was talking about. So these are the myelin pieces. So these are actually what are called Schwann cells, and then there is little gaps in between them called nodes of Ranvier. Okay. So those Schwann cells are what are actually responsible for producing that myelin. The myelin is just kind of this jelly roll like insulation that's around the outside of the axon itself, and it's there essentially to help speed up this process. If you guys remember in bio, if you had either Mr. Vladico or myself, you probably watched Lorenzo's Oil. And in Lorenzo's Oil, Lorenzo had a disease that was actually causing this myelin to be worn away, essentially. Okay, these are the gaps. Then the gaps I talked a little bit about just a few minutes ago. Those are the nodes of Renvier. Those are just the little spaces. If I go back here, those are the spaces in between each one of the Schwann cells. Okay. Cell body itself, it's got a couple different pieces in it. So it's what's referred to as the Nissl substance. This is a specialized type of endoplasmic reticulum that's inside of the cell body. And then there's also neurofibrils. If you guys remember from cell biology, we just talked about fibrils. Fibrils essentially are the cytoskeleton, helps that cell body maintain its shape. Okay. Other pieces then inside of the cell body, the nucleus, which is the large dark center, and then the nucleolus, which is the center of that. Okay. Basic functional properties of neurons. So there's two. There's what's called irritability, so the ability to respond to a stimulus, and conductivity, the ability to then transmit that impulse on. The plasma membrane on the outside of the neuron is polarized, meaning it's positive on one side and negative on the other. And because of that, when you depolarize it, that's what causes this impulse to then be passed from one cell to the next. All right, so starting a nerve to a nerve impulse essentially is a depolarizing of that membrane. All right, you can see this is at resting. You've got positive on the outside, negative on the inside. This is essentially done by two ions, sodium, Na, and potassium, which is K. When you swap those two places, all right, what will happen is that will stimulate this to essentially depolarize along the length of the fiber. And then once it reaches, then it'll repolarize itself. So depolarization simply is allowing sodium ions to flow in, right? And then once those exchange, they propagate or start or initiate this action potential because they're swapping places. So sodium's positive, it moves in, forces that negative to move out and cause this depolarization to kind of move along. This creates what's referred to as an action potential. If we start an action potential, it's going to propagate over the entire axon. It can't stop. So what happens then is potassium ions then swap places with the sodium. So sodium's going in, potassium's going out. And then that causes the membrane to repolarize as soon as it depolarized. Once all that's taken place, then we'll activate our sodium potassium pumps that are located within these cells, and then we'll swap those two back to where they're supposed to be. This requires ATP to be able to do that. So to repolarize a nerve cell, you have to have ATP available. If you don't have it, then it will not repolarize. It's the same thing with a muscle fiber. So if once we depolarize it, if we don't have enough ATP available, it will stay that way. Okay, so this just talks a little bit further of nerve impulse propagation. So the impulse will continue throughout the entire cell body. And then obviously we talked about this just a few seconds ago. So the impulse moves faster in cells that have myelin than cells that don't. All right. Continuation of the nerve impulse. So the nerve impulse can cross the synapse to another one. So once that nerve impulse hits the axon, it's going to cause a neurotransmitter to be released that will then go to the next one and then be picked up by the dendrites of the next neurotransmitter, not neurotransmitter, the next neuron, and then stimulate that whole one to depolarize again, and then it will just keep that same basic process going up. So you can see that a little bit here. So this is our nerve impulse moving along, hits the end, causes the release of these neurotransmitters. The neurotransmitters pass the gap and then are picked up by these little receptor sites. All right, those little receptor sites are can be blocked by using medication. So people who have migraines, they a lot of times have an overactive nerve propagation that's causing that neurotransmitter to be released. Well, the medication that we use could block that neurotransmitter's absorption and then not allow it to be released. So it would not allow the next nerve to be propagated there. Okay. All right, we'll go ahead and stop there. We'll pick back up with some brain parts tomorrow.